Hello, everyone. Thank you for joining us. Just let everyone filter into the room here and get connected to audio. Thanks so much for making some time this morning to come and chat with us or whatever time it is, wherever you are. A couple of housekeeping things just as we're getting going. This is meant to be a chance for us to show you a little bit and hear any questions that you all have. So feel free to unmute yourself and come on with a question when you're ready. If you want to turn on your camera and let us see your pretty smiling face, then feel free to join us. Otherwise, totally understand. So we've got a couple poll questions. Just as you join the room here, feel free to answer those at your leisure. With me this morning, I have our Chief Science Officer, Christy Lawton, up there at my top left. My name is John Harkness. I'm the CEO of Rewire AI. Really excited to show you our latest developments here. We've been putting a lot of work into Polygon here for the last while, so it's pretty exciting to show you what we have. So, all right. Looks like we've got a number of people answering the polls, so awesome. We'll maybe just leave that up here for another second, kind of as people come in. But yeah, so I see a number of familiar faces here. If you are a former Pipsqueak user, thank you for coming and joining us to hear about Polygon. If this is your first time checking out Rewire and our image analysis software, then welcome. We have been building Pipsqueak/Polygon for about the last almost 7 years now. So this has really come a long way since its early beginnings. Pipsqueak AI started when I was a postdoc at Washington State University. If you're one of our very first users, then you remember using Pipsqueak in Fiji/ImageJ. And it's a fantastic tool for doing some image analysis, and we've come a long way since then. What we're talking about today is the release of Polygon. Just full disclosure, Polygon is basically what you've experienced with Pipsqueak. We rebranding with a brand new name. This is what we're calling version 3. So really built on the exact same tools, and I'm going to show you a lot of that. So I think most of what you're going to see today is probably familiar if you've used Pipsqueak before. So yeah, just a little bit of a roadmap, what we're going to talk about today. Start off, I'll just show you kind of how to get going with Polygon, especially how to transition over to Polygon if you are a Pipsqueak user. And then we'll take a look at just a couple different of the features and functions inside Polygon: single channel analysis, multiplex analysis. We'll talk about then how to customize some of the AI models to be specific for the types of images and staining that you're bringing in. So again, feel free to ask questions throughout. We have a chat open up here, so if you want to type your questions in, I think Christy is going to be watching that a little bit. I'll try to answer as we go. Otherwise, I'll come back at the end and we can answer some specific questions in our last few minutes. So Pipsqueak has been around for quite a while. We have somewhere around 60 citations of this initial method that we published back in 2016 under the authorship of Slaker et al. So if you're looking for kind of where this came from, that's a really good place to start. A lot of what Pipsqueak and Polygon are really meant to do is to standardize the methods of image analysis that we had started to develop in Barb Sorg's lab at Washington State University. So what we're showing in that paper are really some of the concepts around what we're standardizing, how we actually see that improving the intra-rater and inter-rater reliability of the analyses that we're doing, and then hopefully how that can also impact your quantification. Where Pipsqueak and Polygon have gone since those early times is now we've built in a lot of the AI tools that really help to automatically detect these targets in a reproducible manner. So hopefully you'll find that this is also really impactful for your quantification. So, yep, check this out. On our website, we actually have a number of citations available. So if you navigate over to rewire.ai, go over to our resources tab, and down at the very bottom, you can find a list of citations. A lot of this is based on that initial publication, but you can really follow it from there. Additionally, I'm going to show you where to find some of the other stuff on our website here. So with that, Polygon is a really big change in how we are doing our image analysis. And probably the most important thing to mention right off the top is Polygon is now free. What we decided is that, you know, the image analysis capability that we're providing here is something that's really been baked into a lot of research workflows. We think that's really important for the reproducibility of that work, for others to be able to then replicate those results. And so we decided that this is something that we want to make available to the community, really going back to our early roots. So just about everything that you want to do in Polygon is now available for free. If you're a power user, if you're doing some of the more advanced things that Polygon's capable of doing, then we also have an additional package that makes those functionality or those features possible. And so I'll show you a little bit more about that when we get there. But takeaway here is if you are using Polygon for your analysis, you're probably in a great spot to use our free tools. So let's dive into it. I'm gonna show you how to navigate a lot of the functions in Polygon. But to start off, let's go back to the website here, and I'm just going to show you where to navigate to really get going with this. I'm seeing from the polls that we've got a lot of current users, but maybe about half of the people haven't tried this before. So let me show you where you can find it. So on our website, rewire.ai, you can navigate over to the Polygon tab. We've built Polygon on a platform that enables it to be, our AI to be integrated into a lot of different capabilities. So we have a focus for some of our commercial partners, some of our enterprise partners, where we're actually able to integrate our AI into their systems. If this is something that's interesting to you, please feel free to reach out to me. I'm going to pop my email in the chat a little later on, or maybe Christy can pop it in now, because we have some really great tools that make this AI something that can, you know, provide further capability for your devices or your software. For everyone else, you can head over to researchers or head over to Polygon. And what we've got right at the top is just the ability to download this right onto your computer. So Polygon is an application that's going to live on your desktop or on your computer. So I'm on a Mac, I would download that. And then from there, this is a normal installation process that you've experienced from anything else you download. Some security protocols that you'll probably encounter during the download process are, you know, just to verify that you do want to install this on your computer. It's something you're getting from the internet. So these are things that you should expect to see. It's typical to see that. It's not anything to be concerned about. But do feel free to reach out to us if you have trouble with installation. So from there, Polygon opens up pretty much the same way that you see Pipsqueak open up, and something that you're probably pretty familiar with. So the very first thing that I'm going to see when I open up Polygon is I'm going to see the screen for me to log in or to register. And so rather than close it here, I'm just going to sign out. So great. This is probably what you're seeing if this is your first time. If it is your first time, head right over to create an account. This is pretty straightforward. All you need to do is enter your name, email, and password. That email address is going to be used to send you a link to verify everything and get logged in. And this is an important step because Polygon is a registration, is a login that you can use on any computer you install it on. So if I go onto my work computer and log in with my email address, I'm going to be able to pull up my same license. So this makes it possible for you to work anywhere, really easy to transition back and forth, but that's why you register. So here I'm going to log in now. It's always scary to try to remember my password in front of people, but I think we got it. Cool. So there we are. Another thing that we enabled in here is actually two-factor authentication. So really cool if that's something that's important to you. So right here from the top, we can see our Polygon interface. On the left, I've got a number of the projects that I've started. These are all the different image categories and experiments that I've put together. On the right, I've got some of my analysis tools, and in the center, you're going to see our image panel. Before we get to that, I'm going to come back over here and I'm going to show you just a little bit more on our accounts window, because this is where you can go and set up some of those password changes, things like that, and also hop in here and check out our premium features if these are things that are important to you. And probably what's most notable here is our batch image processing and the ability to train as many custom models as you would like. So even in the free version, you can train 2 custom models. This is what we see being sufficient for the majority of our users. But if you're a power user, you want to go beyond that, then you can add on some of these additional capabilities and really take it from there. So the other thing that's really useful in Polygon is we have a really nice help and tutorial section. So right up top, I've got access to some tutorials that are actually going to walk me through some of the functionality in Polygon. These are interactive tutorials that might show me how to custom or to build a custom model, or to do single label analysis, or just where some of the buttons are. So if you're getting going and struggling, have any questions, this is a great place to start. Additionally, our very own Christy Lawton put a ton of work into developing this really fantastic how-to document that is going to answer a whole lot of the questions you have. From our FAQ section here, you can download the PDF. This is something that you might print out and have with you, or just have on another screen. That's really going to walk through just about everything that you would want to do in Polygon. So really powerful resource. Feel free to check that out. And so, yeah, lastly, a really, I think, powerful thing to know how to do is to use some of our hotkey functionality, especially as you become a power user. A lot of these keystrokes are going to speed up some of the workflow and just things that you're doing. So feel free to peruse that on your time and check it out. Great. So let's go ahead and dive into just a little bit of Polygon's functionality. So I have a number of projects that I've previously created over here. I'm going to go to this one that I created for today that's stock full of a whole bunch of imaging that I did actually back in my postdoc. So, again, if you're a longtime Polygon user, then you're probably familiar with some of our initial stuff looking at Paranormal Nets. This is some of the WFA staining that I did on some Paranormal Nets back in my postdoc. And so, these are really fantastic images to analyze. I think it really shows off Polygon's capabilities, because one of the hard things about images like this are the definition of where a net starts and stops is sometimes a little bit tricky. And we have a ton of overlap of these cells. So what I can do here is I'm going to go into my model library, this dropdown over on the top right of my image analysis panel, and I'm going to choose the WFA model that I know works pretty well on this. This is where a little bit of experience with Polygon makes it pretty easy to find what you're looking for. And I can change a couple of the detection sensitivities, things that are going to make the detections a little bit more specific for what I'm looking for. And then finally, when I'm ready to go, I can press detect cells. And so, oops, I've already got some on there, so I'm overwriting it. That's fine. So what you're going to see here is in real time, this image was analyzed. It's found a number of paranormal nets in here. It's found a lot of WFA staining. And for the most part, looks really good to me. But this is where I think our workflow is really important. This is something that Rewire really believes in, is that we don't want to blindly accept the detections that AI is giving us. We want to make sure that we've had a chance to verify it, double check it, change anything that needs to be changed. And so, here right off the top, I see, I'm going to zoom in here. So I'm going to press Control, oops, and I'm going to scroll in on my Mac. And I see, maybe there's one cell here that actually has 2 labels on it. So there's a couple things that I can do to clean up this sort of thing. The first is just to click on one of those and delete it. Pretty easy thing to do. Or maybe if I don't like either of them, I can delete them both. I can redraw it. I can even switch over to a different shape like a circle, or one of our freehand tools, such as this guy here, which is going to allow me to draw a freehand circle if I want. Or brand new, we've got the ability to paint these circles with kind of a paintbrush tool, or paint these polygons with a paintbrush tool. So really nice way to start to edit all of these detections that we're making. So I'm going to delete these little guys out there, go back. Great. I like that a lot. The other thing that I can do here to start to clean up my image is just change my detection sensitivity and my overlap removal. These both are pretty self-explanatory from what they're called here, but detection sensitivity is basically how permissive the model is for what you've asked it to find. So the more sensitive I go, the more objects I'm going to see. The lower down I put the sensitivity scale, the more stringent the model is going to be in detecting objects. Because really the way AI works is it's building a probability of everything it's looking at. It's deciding how likely is this the thing that I meant to find? And if, we're above 15% sure, then it's going to, or I'm sorry, if it's at least, in this case, if it was 85% sure of what it's looking for, it's going to show me that detection. So I usually run this at about 75%. I think that's a good spot, something to play with on your own. But I am going to show you the overlap removal here, because what I can do with this tool is I can start to tell the model to not show me as many objects that are overlapped. This is something that's going to vary a lot on the type of staining you're doing, your imaging conditions. If you have a lot of cells that are actually overlapping, I'll show you a good example of that in a minute, you might want to leave this pretty low. In this case, I'm going to turn my overlap removal pretty high, and what we're going to see is that's going to remove a lot of those double detections, which is what I'm looking for in this case. So, that's a pretty good way to clean this up very easily. So great, there you go. That's pretty much the end of that analysis here. And I've got a good question. Is it possible to determine density of fluorescence of cells in Polygon? I think I'm gonna answer that in a second, and let me know if I don't answer that in the way that you're looking for. But really what Polygon is designed to do from this point is to go through our image quantification really based on those early Slaker 16 methods. So I've detected all of my cells in a way that I feel is accurate. And lastly, what I'm going to do here is I'm going to approve these ROIs, which kind of goes through and stamps those into records. So we've now saved these ROIs and the quantification of them. And it gives me a nice little check mark over here on the image that I've finished analyzing. So that's a good way to keep track of what you've gone through and analyzed. And so this is taking just a second because of my bandwidth. But once I've done that, I'm going to go ahead and I'm going to export my results. And what that's going to give me is, that's going to give me a chance to go through and see exactly what we've done here. So I'm just going to put this in my downloads, and voila, we've got our results. And Zoom is in my way, but we'll see if we can open this. All right, so I've opened my comma separated value file here in Excel. This is how I like to see CSV files. Definitely open these in any sort of spreadsheet software that you're using. But a lot of information here. Right off the top, in our first column, we're seeing every cell that I detected in this image, and I'm getting sort of an ID number for each one of those cells that tells me a little bit about it. The first part of the ID number tells me the model that I used to detect those cells. It tells me the shape that I used to detect those cells. If I had hand-drawn any of those here, it would be telling me a little bit about that as well. And then from there, there are a whole lot of different metrics that come out of this. We have some really good explanations of that in the how-to documentation. So this is probably something that you can go back through and check out on your own time. But the one that I like to point out here, because this is what we tended to publish in the SORG lab, is our intensity scores. And so what I have here is both a mean background for my image, and then the intensity. And what I like to do is take the mean minus the background score, which in our lab we interpreted it as signal, and report that. And so what I'm seeing here is this is now the quantification of the intensity of that fluorescence. So going back to that question, I think this might be what you're looking for. This is a fantastic way to quantify intensity. You can also look at integrated density, and this is maybe a little bit more of a relative metric of how large that ROI is. So things to take a look at on your own time. Again, check out the how-to documentation. But as a rule of thumb, everything that you're seeing in here in terms of quantification is something that you're probably familiar with seeing in Image J. we're using a lot of those exact same techniques, a lot of that exact same math. So what you should see is that these quantifications are what you would expect from that type of analysis. So great. Yep. Next question there, Andrew. Thanks. We've got the XY location. This is actually based on the center of mass of these ROIs. I'll show you a little bit more about that in a second. And can we manually set the XY origin? In this case, I think, maybe the best explanation for that is that, we could be moving the ROI, or what I think is really important to show here is we can do some stuff with background subtraction. So this is, I think, a really good way to, again, compensate for any background staining, any background fluorescence that you're seeing, and then really just pull out signal. So with my initial background subtraction selection, I've applied a rolling ball to this, and I have applied a measurement of background fluorescence. This is something that kind of gets into the weeds, so I'll point you back to that how-to document. We have a really long section on how background subtraction works and the different ways that you can manipulate this. But one of the really nice things to show here is that I can, I can, select some of these ROIs, and I maybe can move them, or I could delete them if I don't like them. This is where I could go in and maybe just draw my own background sections if that's what I'm looking for. Again, that grid of background measurements that we take is based on some standardization that we did in that Slaker 2016 paper. So check that out if you have any questions. Great. So back to this. And then can you quantify the size of each cell ROI? Yep, another really good question. So once I've applied the background subtraction to this, what we're actually going to see in our export is just the signal above background. And that's because the program goes through and it sets everything that's below that background threshold to be not a number. It's a really great way to remove those from your averages, from your means. And so what we're going to see here is when background has been applied, my mean intensity is actually just going to be above background, and then my mean minus background is just going to be signal above even that that low level background level. So again, something to check out in the how-to documentation. So great, Andrew, you had a question about 3D. I'm going to come back to in a second. And then Anne, is that per surface area, a long dendrite picked up in a rectangle has a larger body? Yep, than the cell body. So I think that last thing I said there is probably a bit of an answer for your question. So when we subtract out background, we're just looking at fluorescence above NaN. So that's going to be either just your intensity or your intensity minus background. So great, that's kind of a really quick introduction to everything in the in single label analysis. This is super powerful. I think one of the very first things that you're going to want to do in single label analysis is start just playing with these settings, finding what works for you. And one of the things that you're probably going to want to do when doing that is find the correct model for your type of imaging. We have a really large library of pre-trained AI models. These are all models that we've developed in-house. These are based off of data sets that we've hand-checked and hand-annotated. So, we feel really confident about what these are producing. But of course, these are all trained off of some particular image format or, image type, staining method, all of that. So it might differ a little bit from what you're doing. In this case, this is a parvalbumin image, but I might not really know which model is going to work very well for that. So we have a really good tool here called Recommend a Model. It's the very top thing in your library. And when I press Recommend a Model, what's going to happen is it's actually going to run that image against a number of the different models in my library. And so when this is happening, it's going to come back and it's going to say, maybe the Parv model is the best choice for this image, or maybe something else is. And that's going to at least allow me to really quickly visualize a lot of the different options that I have. So here, I'm just going to refresh my screen real fast so that we can see those results come in. So great. I'm going to go back to recommend a model. Boom, recommending the model. And so, yeah, what we'll see here is it's going to bring that up here really quick. Cool, there we go. So it's running this image against 6 different model options. In this case, these are some of the more foundational models that we have. These are all object detection models. Polygon now also has the capability of running segmentation models. And so, this is a little bit into the semantics of AI, but, really what we're seeing is just the difference of a bounding box versus a mask that's being put onto an image. And so, here we're looking at bounding boxes. Again, I think this is really good for a lot of different types of imaging, but segmentation might work better for you. We do have a segmentation model available here, so you can try that one out if it's a good option for you. And what you're going to start to see here in the next couple months is all of our models are going to have segmentation capabilities built into them. So you'll be able to switch back and forth and say, oh, I want to look at parallel-Buhmann with segmentation or just with bounding boxes. So great, I've run this model to see which one works best for me. In this case, I have a lot of different options that work really well. If this was maybe a CFOS image, I could just say, okay, great, I want the CFOS model to run on it. I can select the model, and basically that's already done the analysis here. So that's a really good way to get going. Try out that recommend a model function. All right, going back to the questions here. Yeah, can projects be exported and imported back into Pipsqueak and Polygon? That's right. So right off the top, one of the things that you're going to see if you're a Pipsqueak user and you log into Polygon for the first time is everything's going to port over automatically. So the very first time that you open Polygon, you're going to see all of your Pipsqueak projects here present. This should also bring over all of your results. Everything is going to be just the way that you left it. So in terms of publication, if you've started your analysis in Pipsqueak version 2.5 or higher, transitioning to Polygon should not be an issue for you at all. We always recommend just sticking with the version that you're on, but in this case, it is just really a naming difference. So you should see everything present here. You should also be able to see some of the custom models that you've started. Certainly, if you've trained any custom models that are already available in your model library, those are going to be present here. So everything on your account is going to be just the same. If you encounter a synchronization request, there might be a pop-up that says you should synchronize your project. That's basically just checking in with the cloud to make sure that everything is in order the way it expects it. So running that synchronization function should be just fine. If you have any questions on that, if you run into any problems, do reach out to us. Happy to make sure you can get going with it, okay? So great, we'll go back to my projects here. Do we have a model for analysis of bright field phase contrast? Yeah, so non-fluorescent imaging. Yeah, excellent question. And that's also a good segue into some of our custom model capabilities. A lot of the models that we have in our library of base models are trained off of fluorescence. But what's really nice about that is basically the AI is looking for shapes. And so there's a couple tricks that we've given you to potentially do that better. I don't think I came equipped with like an H&E or a DAPI stain here, but what I'm going to show you is down here in our image view settings and in our image analysis settings, we have a couple tools, including this invert image function. And so when I invert the image, oops, I think it's not going to let me invert it because I've already put detections on here. But what that's going to do in like a DAB stain is actually make all the dark cells white and all the background dark. So it's going to look a lot like a fluorescent image. This is using an ImageJ reverse function. So this is just exactly flipping the pixel intensities. You should feel comfortable doing that. And what it allows you to do is use some of the fluorescent models to do your detections for you. Once you've done that, then I think it's really good to take all of your detections and we can bring them over into a custom model and train your custom model there. So that's a great way to continue to build out detection capabilities that might not be present with our base models. Great. So in ImageJ, the image illuminates in different intensities so that we will analyze the intensity at different fluorescent intensities. Is this also applicable in Polygon? So yeah, I guess I'm not totally sure about the question you have there. So feel free to hop on if you want to chat about it, or we can maybe go into this at the end, and I'd be happy to even look at what you're talking about. But yeah, good question there. And then for the UX, is it possible to pan once you've zoomed in? It totally is. That's a really good question. So again, I'm holding my control key and I'm zooming into an image. And then once I'm there, I can hold my control key and I'm just mousing with my finger on the mouse, or however your mouse works, and you're able to pan around. So yeah, some nice tools there. I really like using this image fit to window function, so I can always zoom back out and it's going to maximize the image to my window. That's a really good way to kind of move around there. Yep. So hold control. Yep. Again, a lot of those hotkeys up here at the top. So if you ever feel lost or forget some of the things that we've talked about, great place to find it here, especially this Control T. I use this a lot just to bounce back and forth to be able to see the detections, so I can actually see what's under a bounding box or under a mask. Awesome. So Alex, thank you. That's a really good segue into colocalization. So yes, one of the things that Polygon has really been built for, all the way back to Pipsqueak, has really been built for is colocalization. And honestly, this is because when I was a postdoc, we did a lot of colocalization analysis, and I found it so difficult to go back and forth and find where two bounding boxes overlapped. You know, multiplexing two stains is very hard. I think trying to colocalize 3 stains by hand is almost impossible. So the way that we do that is we allow you to open up your colocalization. In Polygon, there's no limit to the number of stains that you can colocalize. So if you bring in 10 stains or something like that, you can still colocalize this here. I'm going to show you with 2 stains just to reduce the complexity of it, and we can go through it pretty quickly. But in this case, I've got a number of images here. So this is some CFO staining with, again, it looks like some paranormal nets and some parvalbumin that we had already looked at here. And I've got some CFOS in this. So this is a nice image to really see a lot of different things that are going on. So what I'm going to do is I'm going to choose maybe just 2 of my stains here. So I'm going to highlight the CFOS stain, and I'm going to highlight the parvalbumin stain, and I'm going to right click on these. And when I right click on them, I'm going to go to colocalize. This is one way that I can get into colocalization. I could also right click on the entire folder and say colocalize, and I have some colocalization functions over here on the far right. So a couple different ways to get to this point. Colocalization, great. And so what I'm seeing here is I have my 2 images. I have that parv image that we've been looking at, and I've got my CFOS. And now all I need to do is select the right AI model that I want to use to detect these images. So here's parvalbumin. Over here on the right, I have, I'm going to actually go with DAPI because I know the DAPI model works really well on this image. I think this might actually be DAPI. So we've got that. And then all I need to do is press detect cells over here on the far right. So this is really nice because no matter how many different channels I have, it's going to do the detection simultaneously across all of these. So it speeds up the process a little bit. If you came from Pipsqueak over in ImageJ, then you know how long it can take to go through it one at a time. So doing this in parallel is pretty nice. When we zoom in on these images, oops, yep, I'm again running into a little bandwidth slowness here. But when we zoom into the images, what we'll be able to see is the detections on each. We can go through the exact same process that we did for our single label analysis, and we can correct our detections. And then finally, we can co-localize them. So just out of interest of time, I'm going to refresh my window here and come back to the images. So we're going to choose our 2. Co-localize, great. Parvalbumin, and I like that DAPI. Make my windows a little bit bigger. And detect, awesome. Great, so there we go. We've got all of our detections here. This refresh functionality that I've been using, if this is something that that you want to use as well, I'm pressing Command R on my Mac. Control R is going to work. Or if you head over into the file or Polygon window, depending on which version you're in, you can press the refresh button there. So cool. I have now I have the ability to go in, look at these detections. We've looked at parv. I think that's great. Let's look at our DAPI staining here. And, you know, I think those are great. We're just going to stick with it. So now I want to co-localize. Well, in this example, I apparently don't have a merged image to look at. And I think a merged image is really important for colocalization because I want to see, you know, how those 2 stains are overlapping. And so Polygon has this really fantastic tool, create merged image. that's going to allow me to false color these two images and create an overlay that's going to work for my merged if I haven't created one from the microscope. So in this case, I'm going to go with red and green. That's great. Creates, and voila, this is now something that you can look at and run colocalization for. So Polygon now has a number of different types of colocalization that you can use. Our object-based colocalization is probably what you're most familiar with if you're a Pipsqueak user. This is a really great way to see how objects are overlapping. I have the ability to change how much overlap I'm requiring those objects to have. So, in the case of some of these images, we might be seeing multiple small DAPI cells inside of a large, paranormal net or something like that. And we're going to want to be specific about which one of those, which one of the DAPI stained objects is actually that nucleus. So, you can change that a little bit with our minimum overlap there. I've decreased it, so we're seeing more overlap. And that's, a really good way to kind of hone in on what you're looking for. I've also now got the ability to do object adjacent colocalization. So this is really neat because what this allows me to do is to see a number of objects that might be clustered around a base. And so in this case, a really good use case for this is some staining that we've done with a collaborator for inner ear hair cells, where we might be seeing multiple little puncta stained or objects stained within a hair cell. And so we want to know how many objects are in a single hair cell. And so in that case, I can use my maximum distance to decide how far each one of my adjacent objects are to that central object. And then lastly, I have multiple objects within colocalization. And so this is something that's especially useful if you're doing more than 2 channels of colocalization. Here, I need to choose my base marker. So I'm going to maybe say parv here, run colocalization. And yeah, what we see is this pretty much just looks like my object-based colocalization when I only have 2. But if you have more than 2, this is going to be probably a pretty useful tool for you here. So great. From there, I can approve my ROIs just like I've done previously. And then finally, I'm going to export my results. And I'm going to do it again so I can click that. So what's really neat about this export here is it's actually exporting a whole lot more information that we got out of our initial CSV file. So here's my initial CSV. I'm closing that out. And what I'm showing you now in our plexed colocalization or our export file is we have both of our independent channels. So I've got my parvalbumin and I've got my DAPI. And what I'm seeing is there's actually subpopulations in each one of these stains. So I have the parvalbumin cells that did colocalize with DAPI, and then I have those that did not. And so you can see that between the two, you have your whole population of each stain. And now you can start to work down from there and tease out those different data points, those different numbers that are going to be, the overlap or the non-overlap, whatever it is you're looking for there. So really powerful tool. Again, we've got some really good explanation of all of this in our how-to documentation. So, feel free to check that out there. Really, really great tools. The other thing that my export has given me is actually a little bit of information about each one of my channels independently. So again, if you're a Pipsqueak user, you're probably familiar with this. I'm going to be able to see the analysis from each one of my channels so I can look at those independently. And this is something that I like to point out. And for sake of time, I'm not going to show it specifically, but in my export folder, not only do I have my images that we've been looking at, and I've got my CSV results, but I also have these files that are the name of it and then user ROI zips. And so if you're an ImageJ Fiji user, this should look very familiar to what it might be if you export your ROIs out of ImageJ. And that's because this what that is. So what you can do is you can re-import these images and ROIs into ImageJ, if that's something that's useful for you. And you can actually see your whole experiment there. So something that we've seen users do is actually use Polygon Pipsqueak to do some of your cell detection. And if you have maybe a macro you've written for your image quantification in ImageJ Fiji, you can actually bring these detections into there and then run your process the way that you might be currently. So really powerful tool. It lets you customize any sort of image analysis that you'd be, or quantification that you'd be looking for using the AI to get these detections on there for you. And that reminds me, I want to come back to one thing that I I kind of forgot I was talking about earlier, and that is exporting projects and re-importing them. This is a really powerful tool to share projects between maybe users in your lab, or to move from one computer to another, or just saving versions. So from my project window, I'm going to click this top icon, gets my projects. I've got all the different projects that I've created, and here I can right click on any one of those and I can press export my project. I can choose my destination, and that's great. It's just taking all of those images, all of those results, and putting them into a zip file that I can then bring back into another instance of Pipsqueak or Polygon. So to do that, I'm now just going to go up to my file menu, or on Mac, my Polygon menu, and I'm going to say import project. And I just need to find those projects that we were looking at. Actually, we just exported a couple of them earlier. So, this is a great way to kind of see how that looks. And then I would just bring it right back in. So yeah, great question. All right, going back to the chat. Thanks, Christy, for getting to a couple of these. Let's see here. Is there a way to ID each ROI, like numbers on the image? Yeah, that's a really good question. So there's a couple different ways to do that. One, you've seen in our exports that each one of the ROIs has an ID number. Where you're going to find that in the interface here is to click into one of our images. I'm taking that DAPI image that we've been looking at. And over here on the right side, I've got a couple icons. We've been working in the top analysis icon. Under that, we've got our custom models that I'll show you here in just a second. And then finally down here, I've got my ROIs icon. So from there, I now have a table of all the different ROIs in this image. You can see as I scroll over this table, it's responsive in the window, so you can see which ROI you're looking at. And then vice versa, if I'm to highlight one of these ROIs, I can see it over here. This is nice. I can unselect this particular ROI if I want to see it go away. This is not the same as deleting it. This is a nice tool to maybe just see what's happening there. But if you do want to delete your ROIs, particular problem instance, you need to highlight it, delete it, or check this out up here in my tools. I've got my select ROIs tool. So now I can go through and I can highlight a whole bunch of ROIs. Oops, I'm going to highlight a whole bunch of ROIs. And I can delete those all in one fell swoop. So that's really nice. Again, if you've been using Pipsqueak in the past and needed to do a whole bunch of changes at once, it was kind of tedious. So this should speed you up there. These numbers correspond to what you see in our menu. So here, we were looking at our DAPI ROIs. I just deleted a bunch of them, but for example, here's one that's numbered 00136. We would be able to find number 136. Oh, that was one I deleted over here in our menu. So really nice, especially with colocalization. If we open up these two windows, what I'm going to see is, we've got our the colocalization that we saw in our merged image. That's a really good way to go back and see maybe actually what's colocalizing and investigate a little bit deeper into your data set. So yep, awesome. RNA Scope images. I love that. Yeah, this is something that's really interesting. A lot of users have been talking to us about this, and what we've seen is absolutely it's possible to analyze those images. Where you're going to want to take it is probably into the custom model generation. And so that's a really good segue over into this tool. So on my right side, I have this little chip icon. That's my custom models tab. And basically what this is going to allow you to do is to fine tune any of the stock models that we have available for you in the library. It's going to do it automatically. There's no coding or CS experience required here. Because essentially what we're allowing you to do is to go through and analyze your images. And so in this case, I've already analyzed a whole bunch of these WFA images. These are just some examples. Oops, those, yeah, anyway. So I've analyzed a whole bunch of them. I've approved the ROIs, and now I can go to my custom models, and here's the model that I just started for these. And so what we see here is when I start my custom model, I'm going to go back and show you how to start it. So I can press create a custom model and give it my little name, a little description, and then I'm going to associate it with one of the projects that I'm working on. So in that case, it's this webinar project that we're working on. I even here have the ability to share this model. So once it's trained up, it's going to automatically show up in my library. I can also share this with other premium users. So if Christy is a premium user here, that's going to allow us to share that model and both use the same capabilities. So really nice feature there. So back into my webinar model, what I can do now is I can add all the images that I've already approved from my project. And probably the most important thing to think about here is we're training a model really the same way you'd train any animal in a behavioral experiment. We want to be very specific and very consistent with what we show it. So if I'm trying to build an RNA scope model, or in this case, it looks like I've got a bunch of Paranormal Net images, I want to make sure that I'm not only showing it really good examples of the annotations that I'm looking for, but also that I'm only showing it that one class. So if you have multiple channels in your RNA scope, or if I have, as I do here, multiple different types of stains, I want to make sure that I'm not including those parvalbumin or DAPI stains in my model. I only want to show it the WFA images in this case. I can do that a couple different ways. I can go into my gallery view here and I can look at all the images that I'm including. So, that one's good. But this one is an 8-oxo image. This is a calcium, an oxidative stress marker. So I don't want to include that in my model because it's not the same as WFA. So I'm going to deselect it. From there, I can add all those images, and that starts bringing together all the annotations that we've already gone through and carefully approved. In this case, we only require you to bring in 5 of these images of about that size. This is a little bit of a squishy point because the more images you show it, the better your model is going to train. 5 is really kind of a baseline. If you've got 31, as I do here, I totally suggest training on all 31, as many as you can. But at least from a really baseline level, you can start pretty low. And then lastly, I'm going to draw a couple contours within these bounding boxes. So this does 2 things. One, it allows me to see maybe where I was a little sloppy with my my ROIs. So, so I've selected my ROI. I'm going to, I'm going to move my ROI down so that it better encompasses that whole cell. In this case, maybe I'm going to bring in that side of it. It's drawing a little as I do. And then I'm going to draw a contour around that cell. And really what I'm trying to do here is I'm trying to be as careful as possible to capture that cell. And actually, I'm going to delete that because I don't like what I did. I want to capture that cell or that staining right on the edge of the staining. And so this takes a little bit of work, certainly somewhere that you can spend as much time as you can, be as careful as possible to really get the edge of that cell. And then I'm going to press approve. And once I've approved the contour, I can move on to the next one. Again, I only need to do 20 contours like that. The more you do, the merrier, but 20 is our base requirement. And so that's great. Here's another good example where I wasn't very, I wasn't very careful with my ROI. I'm going to change the ROI a little bit. And again, I can, draw the contour around that. One thing to remember as you're going through this is there might just be some really bad examples of cells that you don't want to include. And so there's a couple of things you can do. One, we can actually delete out that ROI. We could redraw it, or I'm just going to skip this one entirely. And I don't want to include that in my data set. That's, totally reasonable to do until you get to all 20. From there, your train model button is going to illuminate and you can press that. It's going to come up with a little warning saying that this is about to train your model. Our model training process is done locally on our server, and so it's something that runs really fast compared to what you might have experienced in like NIS Elements or something like that. But it does take anywhere from a couple hours to a couple days, just depending on the workload. So what you'll see after you press train a model is back over here in your custom model tabs is you're going to have a couple of these that are in training. So you'll see an icon or an hourglass icon on that. Or if it's finished training, you're going to see it up here in your deployed models. Or if it's something that you're still working on, you've got the edit button here. And so I can always go back in and edit those models. So that was a really long-winded answer to yes, we can do RNA scope, and this is probably how you want to do it. This is also something that I really encourage everyone to do to customize models to be more specific for your data sets.
John, there was one other question right after that one about creating an ROI only analyze part of the image. Do you want to quickly show the
great question.
exclusion and inclusion settings?
Yeah, so here's a really good busy image. This is maybe an example of somewhere where I would want to just be analyzing part of it. So I'm gonna turn off my ROI so we can just see part of it. And so I can do 2 different things here. Down from my image analysis settings, I have an area selection tool. And so this is great if I want to say I only want to analyze right here in the center of my image. I'm going to draw that center, press set. Oops, I'm sorry. Press set and then draw my center and now save. And so essentially what this is doing is cropping out the rest of the image. I mean, really, that's all it's doing, is it's cropping out the rest of the image. Now what's going to happen is when I apply my model to this and detect, you'll see that it's just going to detect here in the center. What's kind of cool about this is I've cropped it so we're only seeing the center. Once my ROIs return back here, I can actually go back and uncrop it, so I'll be able to see the rest of the image present. Gosh, my bandwidth is really being problematic for me. But yeah, that's essentially the way that you'd want to be doing that. And see if I can find that one again. Oh, well, yep. So that's going to be the best way to do that. The other tool that you have at your disposal is the area exclusion. This is essentially just the opposite. So here I'm going to press set, and I'm going to go back to my freehand. I'm going to say, like, I don't want that middle area for what, or, here's a good example. I don't know, there's a hole in my tissue already. I don't want that hole, or maybe I don't want these two areas. I'm going to save. That's going to remove just those two areas. And so now again, when I analyze, it's going to remove just those two. So yeah, great question. All right, it looks like the model training relies on contours of a single cell. What if we want to look at circular structures of a wide range of sizes? Yeah, that's a really good question. Essentially what you want to be showing it is just, identical to what you want it to return back to you. So if you have a lot of different sizes of cells, what you can do is certainly show examples of all those different sizes, drawing structures that are a little bit more or harder to find can be problematic with, those WFA cells, those Paranormal Nets. It's a little bit hard to draw just the contour of that cell, but this is where your best guess is great. And this is also somewhere where you can continue to iterate. A couple of the functionalities that we have coming out here in the future are the ability to specifically iterate on a model that you've started. So if I train this model here and say, well, I don't like what it gave me, I want to try again, I'll see version 2 and then version 3, and I can select which one of those versions I would like to use. The other thing that we're going to have coming online here in the next little bit is the ability to train those segmentation models. So you'll be able to say, object detection actually isn't the best way to visualize this. I want to see segmentation. And so that's something that's coming online. And then lastly, one of the things that we have coming online is the ability to train multiple different classes of objects within a single model. And so this is a really good example of what you're talking about. If you have objects of different sizes, or, for example, we do some H&E staining in one of our projects. And with that project, I want to see both the cells, and I want to see background, and I want to see areas with holes in it. And so what I can do is I can classify each one of those different objects as a different class, so class 1, 2, and 3, and we can train a model that can find each one of those. So watch for that here in the next little bit. It's certainly a way to start to tease apart those maybe different subpopulations of staining that you have. Great. Yeah. So if this takes model training, yeah, if this takes off with my colleagues, institutional premium licenses, absolutely, Andrew. Feel free to just reach out to Christy or I, and I would be happy to chat with you more about that. Certainly have a lot of different ways to bring Pipsqueak in or Polygon into to your institution. And then Maheshwar, great to see you. Encountered an issue with annotating and selecting contours when preparing a model for training. Yeah, Maheshwar, I think probably what you've seen is just a little bit of the carryover from some of the projects that we were working on. Actually, since Grant is here, if you want to hop into a breakout room with him specifically, I think we can probably talk with you in more depth on that project, but great question. And then Andrew, yeah, thanks. 3D Z-Stack. So 2 premium features I'll end with here are our batch processing and Z-Stack processing. Really because they're in some ways very similar. So I have here a whole lot of WFA or Paranormal Nets, WFA staining. And one of the things that I can do with this is to batch detect all of these images at the same time. This is just a nice way to speed up going through this process so that I can then go back and do my approval. So in this case, all I need to do is again, right click on either the folder that I want to batch detect, or, if I want to choose maybe just 3 of these images here, I can just select those 3, pressing Control or Command, right click, batch detect, and that's going to bring those up. For batch detection, I'm only able to choose one type of detection model at a time. So these all should be the same class of staining. So WFA here in my case, and then voila, batch detect. And so what I'm going to see is over here in my project window, these are being processed. The check mark shows up as they're processed, and we see all of those ROIs populate there in pretty real time. So from here, I can go in, I can look at my ROIs, make sure I like it. I'm going to approve those, and then I'm done with that process. So this is a really handy tool if you've got, as I do here, a whole list of images. Certainly a lot of the experiments that we were doing in my postdoc would come away with a couple hundred images. So batch detection is a nice way to speed that up. If you're interested in batch detecting for colocalization or for multiplexing, this is just a slight deviation on the same workflow. What I'd suggest you do is, so here, for example, I've got 3 different multiplexed images. I know that the 4th image or I know that the numbering is the same on each one of these. So actually, let's take the 2nd image here. So this is parvalbumin. I'm going to select each 3 of those, and I can go through again. I'm going to batch detect that way, and so this way I can still do colocalization through batch detection, but I'm going to choose each one of the channels and do those one at a time. So in this case, I would run batch detection 3 times to get through these images, if that makes sense. And then lastly, Z-stacks. Yeah, so what I have here is a bit of an example of some Z-stacks that we've imported. So a lot of the images that I've shown you are coming off of a confocal. The process that, again, we talk about in that Slaker 2016 paper is that we sum our images, we sum our Z-stacks before doing an analysis on them. And we do that for a very specific reason. Summing the pixel intensities is going to show you relative pixel intensities across that Z-stack in your 2-dimensional plane, whereas max projecting those is just going to show you the brightest pixel. So, it's a little bit of preference. Whatever your lab, is set up to do, I think is great. But in our case, we like to sum them. In this case, I haven't summed them yet. I imported my entire Z-stack, and there's a really cool way that we can do that. From your project window, I can go up and I can select add new files, and I'm going to scroll down here to import image stacks. And once here, I can choose the stack that I want to import. Let's see if I can find my, yep, great. So here's a stack that I could import, and that's going to show me all the different files that are in that stack. This is, again, very similar to how you might import a stack in ImageJ. And for what I can do is I can either choose the step size. Again, if you're familiar with ImageJ, this is going to make sense. Choose the step size that I want to use, or I can select pattern selection. This is just using the same Boolean logic that you might be familiar with. So I'm going to do asterisks for a wildcard, and then I'd say like ch, I think 00.tiff was one of them. And so now I'm seeing all of the images from just that channel. So that's a really nice way to then import it. What you'll see is that stack comes in just like we saw here. And now what I can do is I can go through and I can analyze, or I can detect, the cells in this one at a time if I want. So I'm going to use the parv image, and I'm going to detect just on this slice. Or I can go through and I could detect on my summed slice. Oops, this is going to lag on us again. And see, where those cells are across the summed 2-dimensional plane. And then lastly, what I could do is I can go through and I can actually detect across my entire stack on each individual plane. And so let's see if we can do that. Let's try this image here. Let's see if we can do that. What I'm going to do is I'm going to actually select my stack here. I'm going to press batch detection. That opens up my whole image. I'm going to choose my model. Great. And I'm going to press batch detect. And so what we're going to see is that's going to place those ROIs all the way through my stack. And now I can go back and I can see those ROIs. I can also see where they overlap, almost like where they co-localize in that 3-dimensional plane. And so, this is going to take us just a second. So, yeah, maybe something for another time to show, but this is a really handy tool to start to see, how a cell starts and stops through your Z-stack, and maybe how that relates to other cells in your image. So try that out. Again, happy to answer questions on that when you get going with it. So, yep, that's the top of our hour. I have spoken at you straight through this. So, if you have additional questions, please feel free to reach out to us. More than happy to talk a little bit about any other things that you want to hear or see. We also do these webinars periodically, so stay up to date with when the next one is. We'll be sending out some email reminders about it. Come and learn about more functionalities, more features, and yeah, thank you for joining us today. Really excited to get Polygon into your hands, see where you take it, see what you can do with it. So, you know, download it, get going with it, and let us know what you think. Thank you all so much for joining us.